Hi everyone and welcome back to Advanced Higher Biology. Today we're continuing with Unit 2, Organisms and Evolution, and we're on to Key Area 3, Variation and Sexual Reproduction. So we're halfway through Unit 2. I did put up a poll as well to say, what do you prefer? Do you prefer going through this all in one go per key area, or do you want it split up into sort of sub-key areas and shorter videos? Thanks so much to everyone who got back to me on that poll. It was really close. It ended up just 51% of people decided they wanted it all done in one go. So that's what I'm going to go for in this video, as well as the rest of Unit 2. To try and keep everyone happy, what I'll try and do is get a timestamp of each sub-key area so you can go and flick between them all rather than just go through this whole thing. But let's just make a start and get through all of Key Area 3. Remember, you chose this. So, to start off with, in the first sub-key area, we are comparing sexual and asexual reproduction. Now, as with a few parts of Unit 2, quite a lot of this you'll probably already know to some extent, but it's just good to try and refresh your memory and have a bit of a discussion on the comparisons of sexual and asexual reproduction. So first of all, there's an idea in evolutionary biology that sexual reproduction in some ways doesn't really make sense because it takes a lot of energy, it requires two partners, and there's also a bit of a breakup of genetic variation. So one of the main ideas is that first of all, males are unable to actually produce any offspring. All the offspring production is only on females, which isn't ideal. You're cutting out 50% of the population. And only half of each parent's genome is passed on to the offspring. We're going to talk about how this can be a good thing in terms of variation, which is what this keeps coming back to. But in terms of a really strong, successful parental genome, if you have a parent that has essentially excellent genes, in terms of evolution, it makes sense just to pass those genes directly on to offspring through a clone. The idea of then changing them up with a partner doesn't make sense. You're disrupting this parental genome. So essentially one of the big benefits, as I said, is actually this genetic variation. The idea is that all of the costs of sexual reproduction are actually outweighed by the benefit of an increase in genetic variation. So this variation is things that you've discussed even back at the start of National 5, discussing how sexual reproduction leads to greater genetic variation. If you have more genetic variation, you're more likely to be able to deal with selection pressures. So essentially, if you have a large genetic variation or a wide range of genetic variation, that's going to be the raw material for adaptation, for natural selection and evolution to take place. So that means that sexually reproducing organisms have a better chance of survival under changing selection pressures. They have more variation, they have more chances to have alleles that are going to be favourable under these conditions, rather than an asexual population just need to deal with it. They might have good genes and survive, or they might have bad genes and just have no chance and all get wiped out. So throughout this, I'm going to try and just pop up some summary pages here of advantages and disadvantages. So for sexual reproduction, as I've said, an advantage is you have greater genetic variation, and that means there's going to be a greater ability for organisms to adapt to changing conditions, to deal with selection pressures, or survive, for example, new strains of diseases appearing. The disadvantage of that is males cannot produce offspring. If there are good genes in a parent, only 50% of those genes are being passed on. And there's also that random formation of gametes that we'll talk about later as well. Again, further disrupting the successful parents, the genomes of parents. This also ties back to this idea of the Red Queen hypothesis that you might remember from the evolution video. So the whole idea was for the Red Queen, it takes a lot of running to stay in the same place. So for example, if a host is able to develop a new defense against a parasite then needs to develop a new way of getting into that host or disrupting that host. The idea there is that sexual reproduction can actually be used to explain this, or rather the problem of the Red Queen is used to explain the persistence of sexual reproduction, because there's going to be more variation. If you have more variation through sexual reproduction, you have more chances for example, for hosts to develop a defense. And for parasites, they then have other chances to develop a new form of offence to get through this and keep this cycle going on. If all the populations were asexual, there wouldn't really be many changes taking place. So for asexual reproduction, again, there are good sides to this as well. So if you're wanting to pass on those whole genomes from parent to offspring, it's a good way of doing this. Also in terms of speed, one parent, if you think of things like bacteria, one parent can produce daughter cells and establish a colossal colony over time just by that exponential growth of asexual bacteria, for example. Now, as we said, it can be good for you just to continue on these parental genomes. It's of particular advantage if you have a narrow, stable niche. So remember, the whole idea is variation is good when there's a lot of selection pressures, when there's a lot of changing conditions, when there's a lot of disease or parasites. Then you want the variation in order to have a better chance of survival. However, if you are in an environment where you have a narrow, stable niche and nothing is really changing, actually being asexual and just keeping that genome going and reproducing quicker is going to be of more of an advantage to you as well. So again, this is just a little summary again of asexual reproduction. It's important that you just know what's good and bad. Don't just think that sexual reproduction is good and asexual reproduction is bad. They both have pros and they both have cons as well. Just a little reminder as well of asexual reproduction. This is something you'll have looked at probably back in the reproduction part of National 5. But there's ways that plants can reproduce asexually. So you have vegetative propagation. You may have done some of this as an experiment. So going back, you might remember potato plants and other vegetables have tubers where they go and release these tubers underground. Each of these tubers can then produce its own tubers, and that's just a form of asexual reproduction. You can plant bulbs of daffodils. You have various parts of, say, aloe vera that you can they can bud off that you can go and add. And then you have runners from strawberries. So you have these plantlets that take off and trail along the ground and then eventually release their own roots and break away from the parent plant. But these are all asexual reproduction. One that you've probably not came across though is something called parthenogenesis. Now, the idea of parthenogenesis is reproduction from a female gamete without any form of fertilization. So originally a lot of this first got picked up when we had isolated female Komodo dragons in captivity that had always been on their own in captivity, and they started producing offsprings, which doesn't make sense in most of our reproductive biology. So this is a form of asexual reproduction, and this is a form of asexual reproduction that can be done in eukaryotes. It is far more common in colder climates, which are areas which are far, far less likely to have any form of parasites. It's disadvantageous to parasites, or regions where there's just no parasites going on. So again, this kind of confirms what we've said before about the pros and cons of asexual reproduction. It is better to have variation through sexual reproduction, but if you're in a stable, narrow niche where there's less selection pressure through, say, parasites, then asexual reproduction can be an advantage. In terms of the growth of these populations, this diagram just sums up. It's something you've seen before. It is an advantage for asexual reproduction or asexually reproducing populations that they can rapidly outgrow sexual ones. As you can see here, you have just the start of one group here. Through asexual reproduction, they can spread so quickly. With sexual reproduction, you need to find a male and a female. The male is then not producing any of their offspring. It's the female producing the offspring. They need to find a new male. It takes a longer time. So asexual reproductions rapidly outgrow sexual ones. Another idea to think as well, though, is we do talk about asexually reproducing populations as clones, and we talk about the lack of genetic variation, and that is true. They have very, very little variation, but there can be some. So it's important for you to know that in asexual reproduction, the offspring that are produced, there will be some form of mutations that go on that do give some form of variation, which can allow natural selection and evolution to occur. Obviously, this is just in a very, very small way in comparison to sexually reproducing offspring or organisms, but it's just important to know that it'd be wrong to say there is no variation in asexually reproducing populations. There is some, but it'd only be a tiny amount. And finally, something you've came across before in higher biology is the idea of horizontal gene transfer. So some organisms that reproduce by asexual reproduction, for example bacteria, often have mechanisms for this horizontal gene transfer. If you remember, that is the passing of genetic information between members of the same generation. So this can happen in the plasmids of bacteria and yeast as well. And it's just the idea that if you're able to pass genetics or some form of your genetics across to members of your same generation, that will result in much faster evolutionary change because you are getting a bit more variation going on there than organisms that only used vertical transfer. Vertical transfer being from parent to offspring to F1 to F2, that side of things. Okay, we're on to the second out of 3 sub-areas of this part here. We're looking at meiosis. So this is going to be quite similar to the mitosis part that you've seen in Unit 1. We're going to go through the process of how this works, and we're going to look at essentially where variation takes place within the gamete formation in this sub-area. So, first of all, the idea of meiosis is the division of nucleus that results in the formation of haploid gametes from a diploid gametocyte. So hopefully you remember haploid, 1 set of chromosomes, diploid, 2 matching sets of chromosomes. In diploid cells, chromosomes typically appear as what we call homologous pairs. Now this is again something you may remember from the mutations part of higher, but we talk about homologous partners and that side of things. So if you look at this, this karyotype here, this mapping of human chromosomes, you can see homologous pairs all lined up. So homologous chromosomes are the same size as each other, they have the centromere in the same position, they have the same sequence of genes at the same loci or location within these. So if you have a bundle of chromosomes from any organism, you're actually able to separate them into what their pairs are because these homologous pairs are going to look essentially the same. So it's important to know what are these defining characteristics of homologous chromosomes. So same size, same centromere position, same sequence of genes at the same loci. So meiosis itself, there's 2 distinct parts to this. There's a little summary here that you might want to go back to. But what we're going to go through is first of all meiosis 1 in terms of what happens, and then a sort of final splitting that goes on before meiosis 2 so that we end up with 4 haploid gametes. So, first of all, through meiosis 1, we have the pairing of these homologous chromosomes. So, the chromosomes are going to replicate. They consist of these 2 genetically identical chromatids attached to the centromere. This is stuff that you'll have came across before in the producing new cells part of National 5. So, these chromosomes condense, and homologous chromosomes are going to pair up. So, they're going to match with their homologous pair. At this point, something called crossing over takes place. So where there are points of contact between non-sister chromatids of a homologous pair, you're going to have something called chiasmata. So chiasmata you can see in these diagrams here, essentially where the legs of the chromatids touch. If chiasmata take place, then sections of DNA are going to be exchanged. So you can see here through the diagram, we have the capital A's and the lowercase b's, lowercase a's and b's. You can see where the touching and the crossing over has taken place, and we've basically swapped these genes going on. So through this, you have something called linked genes. If you have linked genes, they're the genes on the same chromosome. So on the left, on this white chromosome, you would have A, B, C, D, and E uppercase. On the grey one, A, B, C, D, and E lowercase. Those would be linked genes on the same chromatid. So again, if they cross over, you can result in new combinations of the alleles of these genes. The other point to remember here is this is random. This is not a planned, or you can't go and sort of predict where it's going to happen. This is just where these chromosomes are touching, and crossing over is random. It's going to lead to genetically different recombinant chromosomes. Remember, recombinant basically just meaning a new combination of these chromosomes. So first of all, that's going to lead to a large amount of variation. At this point, again, we're going back to some National 5 terminology that you may remember. You have spindle fibres. They're going to attach to the homologous pairs and they're going to line them up at the equator, and then these are going to be getting pulled apart. Now, the orientation of the pairs of chromosomes at the equator is also random. In Natural 5, when we talked about mitosis, we just showed them all nicely arranged in a little sequence. Where these chromosomes actually line up is entirely random. Now, that might not seem too important, but if you look at the difference between the blue and red chromatids in this diagram here, how they actually separate can have a big impact on the formation of that gamete. So essentially, each pair of homologous chromosomes is positioned independently. It doesn't matter which one's maternal, which one's paternal. There's not the maternal ones are all on the northern side and the paternal is on the southern side. They're all going to line up independent of what's going on. And this area is known as independent assortment. So again, if you look at these two possibilities here of just these two different pairs, depending on what side they're on at the equator and then get pulled apart, leads to a bunch of different combinations taking place. So again, this independent assortment is going to lead to variation. At this point then, the spindle fibres are going to pull the chromatids apart, they're going to travel to the opposite poles, and just like in mitosis, cytokinesis is going to occur. So you're going to have two daughter cells forming at this stage. Finally though, what's going to happen is each of the two cells that are produced during meiosis 1 will then go through division one more time, and again the sister chromosomes are going to get separated. What that means then is a total of 4 haploid cells are produced. So at the end of meiosis 1 you have 2 diploid cells, at the end of meiosis 2 you're going to have 4 haploid cells, which is how you have your haploid gametes. Again, I've just put this as a little bit of a summary here, but the main idea is the two main ways in which variation is produced by meiosis is the crossing over stage, so the chiasmata, swapping those genes, separating linked genes within the chromosomes, and then that independent assortment when the chromosomes line up. And again, that's just random, it's down to chance, and that has a huge impact on what is produced at the end. And finally, we're on to part C, which is sex determination. So for this subkey area, we're mostly looking at the idea of sex chromosomes and how they work and what else can determine sex within organisms. So in birds, mammals and some insects, we have the presence of sex chromosomes. So for example, on this karyotype that we looked at earlier on, you can see all of the chromosomes within this human. Straight away you should be able to figure out though that this person is female. The reason for that is there are 2 X chromosomes on the sex chromosomes, and there is no Y chromosome, which would be present in a male. So if you're aware, females are XX, they have 2 X chromosomes, males are XY, 1 X chromosome and 1 Y chromosome. Now the idea in here is that in most mammals there is a SRY gene on the Y chromosome, and that determines the development of male characteristics. So essentially, if you have a Y chromosome, then you'll have that SRY gene, and that's going to determine the development of male characteristics, and you'll be male. If you do not have that Y chromosome, for example, you have 2 X chromosomes, you will not have any of the development of those male characteristics, and therefore you're not male. This is important for a couple of different reasons here, and this is something you may have heard of before in terms of different genetic conditions or carriers in males and females and how this works differently. So if you are heterogametic, if you have 2 different XY chromosomes, so if you're an XY male, then you lack most of the corresponding homologous alleles on that shorter Y chromosome. You can see on this diagram on the right hand side, these chromosomes are much different in their length. What can then happen here is you can have sex-linked patterns of inheritance, which is found in carrier females and affected males. So for example, in homogametic females, so they both have 2 X chromosomes, one of the 2 X chromosomes present in each cell is going to be randomly inactivated at an early stage of development. So in each individual cell, there are 2 X chromosomes in females. You only really need 1, so one of them will randomly be turned off. That Inactivation, that's switching off those chromosomes, stops any double dose of gene products because that could be harmful to cells. That could almost be like a duplication mutation taking place in every single cell when it comes to your X chromosome. Now, carriers then are less likely to be affected by deleterious mutations on these X chromosomes. But if you remember, the key thing is any one of these X chromosomes can be switched off, can be inactivated, and it's random each time. So because in each cell it is random, half of the cells in any tissue will have a working copy of the gene in question. So that's good, and that's why it's less likely for females to be affected by these, because essentially in your heterogametic males with XY, if you essentially have a dodgy X chromosome, that's the only X chromosome you have, and you're going to be producing that gene, producing that protein that's going to have some sort of deleterious product for you. If you are female though and you have two X's, it does mean that half of your cells, roughly, are going to have that issue, but the other half won't. So it's going to be less of a problem. How we can actually see this is in something called mosaics. So commonly in tortoiseshell cats, you'll see they have these distinctive different colours. The reason for that is in the X chromosome, there is an allele for hair or fur colour. So in these cases here, you can see half of the genes, for example, have like a lighter, more gingery colour that has been expressed, and the dark allele has been inactivated. In the other half, the dark allele has been activated, and the more ginger, lighter colour has been inactivated, showing this kind of pattern going through the body. The other part we talk about is something called hermaphrodism. So hermaphrodites are a species or members of a species that have functioning male and female reproductive organs. So the key point being they both still function as male and female reproductive organs. They produce male and female gametes. In these organisms then, hermaphrodites will usually have to find a partner to exchange gametes, just in the standard form of sexual reproduction as well. The benefit to this, or to these individual organisms though, is that if it is quite rare to actually find a partner, then the advantage of being a hermaphrodite is that partner does not need to be of the opposite sex. If you're a female, you do not need to find a male, or vice versa. You can just find another member of your species, and because you're both hermaphroditic, it means one of you can essentially be the male and one can be the female. The other thing that can determine sex determination as well, though, is it's not always just genetics or the chromosomes you have. In some species, there are also environmental factors. So this is one in which you'll find lots of case studies online. Some of them are really interesting, so I'd suggest you should go and have a look at them, and you may get a case study to read up in an exam as well and discuss what's going on. But essentially what can happen here is sometimes it's the temperature of egg incubation. So depending on what temperature is in the environment, the offspring within that egg are going to either be born male or female. So there's an example here of alligators, that if the temperature is between 32 and 34 degrees Celsius, then the alligators will be male. However, if the temperature is below 32 degrees Celsius or above 34 degrees Celsius, then they are going to be female. So this is all down to just the environmental temperature, so it's an environmental form of sex determination, but it's also something that's causing a bit of an issue, and to put it lightly, because of global warming. Because some areas are heating up more, you'll find that some species are only having a lot more females or males rather than a sort of more 50-50 mix. The other elements, and again, like I said, look into some case studies in these as well, can be that some organisms will have their sex determined as a result of competition or size or parasitic infection as well. These essentially are having an impact on the ratio or the sex ratio of offspring that are being born. And in some cases, this can also be down to resource availability. In some cases, it looks like it's down to if there's not a lot of resources, offspring or parents are not wanting to produce offspring that are all the same sex as theirs, because they can be competing for different resources. So instead they're all a different sex and they can go and spread out and basically have less competition going on. So there's more that just determines sex in certain species than just having the sex chromosomes as well. And like I said, I'd encourage you to go and read some case studies of these. So that is all for this key area. That's another one down. If you've managed this all in one go, well done you. If not, you've been flicking through this. Hopefully you found this all very, very helpful. There are only 2 to go now as well. They are quite long, but they are pretty interesting. And hopefully you're finding these videos useful for your revision. If you're watching these as they come out live, it's not that long now until the exams. So keep it up. It's a lot of content, but go through there and just have a look at the main parts that I'm talking about in here as well. So again, thanks so much for listening. Please pass on to anyone else who is listening to or doing advanced higher biology this year. Hope you find these useful, and I'll see you soon for key area 4. Bye for now.